So this is the final PhD seminar for Haysem Alhassen. Haysem has had to deliver his final seminar in a slightly unconventional way because of a combination of lockdown and a newborn baby in his house that isn't silent during many of the daylight hours, from what I understand. So what Haysem have done and I have done is recorded his seminar in the depths of night when baby was asleep for a couple of hours, and we will be playing the recording of that today, and I'll start that off in just a few seconds. So we'll hope for the technology gods to be kind to us. Haysem is a student that came to work with Fiona, Dyer, and I looked back on the dates in 2017. He is someone who has been one of those sort of vampiric PhD students because he has a full-time job that he also does. He also has a family to look after. And so he spent an awful lot of late nights and weekends working on this PhD project. And during that time, I think we've seen Haysem really develop a new set of skills. He's not someone who arrived knowing it all already by any means. He's worked extremely hard in order to improve his skills. Coming from a chemistry background, he's had to learn a lot of ecology and very patiently listening to Fiona and I wax poetical and at great length about the importance of ecology. And he's also taught us a lot about chemistry. So I think it's been a really neat experience supervising Haysem. I think in his introduction to his talk, you'll get a real sense of how far he's come, and he is very much someone who is worthy of our admiration for all that he's achieved. So I'm going to share my screen with you now, and I'm going to bravely share a video file with you. So we'll start that right now. Welcome everybody to this week's IAE seminar, and it really does give me a great deal of pleasure to introduce today's speaker, who is Haysem Alhassen. Haysem has been working with Fiona, Dyer, and myself on his project for the last 3 or 4 years. He's one of those people who has succeeded in completing a PhD while somehow raising children and also working a full-time job. And in fact, Haysem has a very new member of his family who's joined him within the last couple of weeks. So if he looks a little sleep deprived, it's probably not because of preparing for a seminar. It's probably because he has a newborn baby in the house. Haysem's project is a really interesting one. It deals with some of the issues that we've been really interested in terms of low dose but pervasive doses of pharmaceuticals in the environment. I'm not going to spend a lot more time introducing Haysem's background because I know he's going to give you a bit of an overview at the start of his talk. So I will now invite you to listen to Haysem.
Thank you, Ross. Yeah. Good morning, everyone. I hope you're all doing well and safe amid the current lockdown. And as Ross mentioned, in the last few days, I was blessed with my second daughter. Although it's a bit stressful, to look after a crying baby while I'm doing, wrapping up the final stages of my PhD, yet I really enjoy this stress. I came to Australia in the year 2000 as a political refugee with my mother, sister, and brothers. At that time, I just finished year 12, but I was the older among my brothers, and I had to play father for them. That was a stress. A really big stress. I wanted to pursue my study and get a bachelor degree in science, but I had zero English at that time, literally zero English. And I needed to overcome that language barrier, and that was another stress. I had an extended family overseas who fully depended on me for financial support, which means I have to work to support them. So I had to study and work full-time at the same time, and that was another stress. But the biggest stress, I left behind my father in deep trouble. He was a lawyer and a human rights activist. And as you know, dictators love human rights activists and love to keep them close in a prison. So I was thinking all the time how to save my father and get him to Australia. And that was the biggest stress of all. So you see, multiple stresses as the title of my project, as you see on the screen, multiple stresses. That's why when I discussed my PhD project with my supervisors, and we agreed about this title, I saw a commonality between this title and my life. Today, I'm going to talk to you about stressors that affect ecosystems containing the most important element for all humans, water. So this is the study aim and objectives. So we aim to study the effects of multiple stressors on freshwater ecosystems processes. To do that, we had a number of objectives, and as the diagram or this schematic diagram shows you, pharmaceuticals are part of the big problem or stress of chemical pollution. So we had pharmaceutically active compounds that has been detected in fresh waters. And here we just picked 4 of the common pharmaceuticals in order to conduct our study on. So the research question was actually, how do 4 selected pharmaceutically active compounds influence a critical ecosystem function in stream ecosystems? Specifically, how do pharmaceutically active compounds affect the leaf litter decomposition rate? This is a very vital and essential process in freshwater ecosystems. So how do these pharmaceuticals going to affect them? First, separately, and we had a major experiment for that. And secondly, in combination, and that was the second experiment. And thirdly, combined with temperature stressor as an environmental stressor. So this is the overall thesis structure. I had 7 chapters. Chapter 1, introduction. Chapter 2, literature review. Chapter 3, photodegradation of caffeine and paracetamol. Chapter 4, effects of amoxicillin, caffeine, paracetamol, and sertraline separately on LLDRs, that's leaf litter decomposition rates. And chapter 5, effects of pharmaceutically active compounds mixture on leaf litter decomposition rates. Chapter 6, effects of pharmaceutically active compounds mixture and temperature combined together on the decomposition of leaf litter. And at the end, my conclusion will be a chapter, and that's titled General Discussion. So I'm going to start with chapter 2 because chapter 1 is merely an introduction about the study and how is it going to work and how is it structured. So chapter 2 is what I'm going to talk about now. It is the literature review chapter. There are various pathways or sources of pharmaceutically to the fresh waters. That's why the first question that may come to mind is how these chemicals get to the fresh waters in the first place. So this chapter focused on answering the questions, how do pharmaceutically active compounds enter the environment? What concentrations occur in the environment, and that's the freshwater ecosystem environment? And what are the possible pathways for pharmaceutically active compounds, impacts, and degradation? There are various ways of pathways or sources of pharmaceutically active compounds to the fresh waters. Wastewater treatment plants are considered as a main source of pharmaceutically active compounds in streams and rivers. Agriculture and manure from farming process, where a lot of antibiotics are used, are another sources of pharmaceutically active compounds in fresh waters. In countries where there are no strict waste disposal systems, industries and hospitals can be sources of pharmaceutically active compounds in fresh waters too. Pharmaceutically active compounds are detected in concentrations range from nanograms to micrograms. And they degrade by various processes such as volatilization, photolysis, sorption to particles, maybe hydrolysis, and so on. And this schematic diagram here, also this figure, shows you how do they get to the surface water or to the fresh waters. And these are some of the main pathways. But before we go to chapter 3, Society of Environmental Toxicology and Chemistry, CTAC, organized a workshop in 2012, and that workshop raised the following important question. How important are pharmaceutical and personal care products relative to other chemicals and non-chemical stresses in terms of biological impacts in the natural environment? Answering this question requires an understanding of the biological impacts of personal care, of pharmaceutical and personal care products. To contribute to the effort in having that understanding, my research investigated the potential effects of 4 common pharmaceutically active compounds: amoxicillin, caffeine, paracetamol, and sertraline on leaf litter decomposition rate. These 4 pharmaceuticals belong to different therapeutic classes, but they are common pharmaceuticals. They are prescribed worldwide. They are very common, and their production is continuous until now, didn't stop. And the consumption of these pharmaceuticals actually is expected to increase over years. So I have, for amoxicillin and sertraline, I had the data of how long they stay in the environment when they get to the waters, how long they stay there, for how long do they persist. But for caffeine and paracetamol, I didn't find that in the data, so in the literature. So I had to obtain that data myself. And to do that, I needed to conduct an experiment studying the photolytic breakdown of caffeine and paracetamol residues in surface water, and that's chapter 3. So again, as I said, this chapter focused on answering the question, how long do caffeine and paracetamol persist in under natural conditions in river water? The method that I followed to conduct this experiment, just I'll go through it quickly. Caffeine and paracetamol were obtained from Sigma Aldrich in the US. Stock solution for both pharmaceuticals has been prepared in an ultra pure water at concentrations of 100 milligram per 100 mil. Concentrations used for caffeine were 1.3, 2.6, and 3.9. So 1.3 is that concentration, and that's microgram per liter. So this is the concentration found in or detected in the natural waters. And I use that concentration, and then I doubled that concentration, then tripled that concentration. Why? Because as I said, all the projections shows that the population consumption of these pharmaceuticals are increasing. So that's why we're expecting the concentrations of these pharmaceuticals to increase over the coming years. That's why we tried to know, before that happened, we tried to know what's the effect of those increasing concentrations. That's why we use 3 different concentrations. And this is applied to all of the pharmaceuticals used in this research. And we're gonna see that in the coming slides. So the concentration for paracetamol was also 10, 20, and 30 microgram per liter. The 2 pharmaceutically active compounds were placed in 200 ml vials containing river water and leaf litter for 28 days. The vials were divided into 2 groups, one placed in a simulated day and night 12 hours, and the other one was in just 24 hours darkness. Light was provided using fluorescent lamps, and that type is called T5. Water samples were collected every day to measure the concentration of each pharmaceutically active compound using Shimadzu HPLC. And here we can see the results we can see the 3 different concentrations used for caffeine and how they break down over 28 days. The means with hollow circles are for caffeine in the light, and those with black circles for caffeine in the dark. We can now determine the half-life value at each concentration from the graph. For concentration 1.3, which is the lowest one there, in the light, we see that it reached the half-life in the 5th day, but in the dark, it happened in the 15th day. The same can be seen with the concentrations of paracetamol here, and clearly those the black circles, that's the means, standard errors in the dark, are taking much longer than the hollow circles, which is in the light. So the breakdown is faster in the light, as we can see here. So as I said, in the results are the half-life values for caffeine in streams water range from 5 to 16 days, and that's depend on the concentration because we have 3 different concentrations. The half-life values for paracetamol in stream water range from 7 to 12 days. So conclusion for chapter 3, photolysis is a vital process for the degradation of caffeine and paracetamol in streams. Caffeine and paracetamol degrade faster when exposed to light than in the dark. And now we have the data for caffeine and paracetamol, and based on that, I can start the main experiments. So the main experiment is studying the effects of amoxicillin, caffeine, paracetamol, and sertraline separately on leaf litter decomposition rates in freshwater ecosystems. This chapter focused on answering the question, what are the effects of pharmaceutically active compounds, stressor, separately, so as a single compound, on leaf litter decomposition rates. The method for this experiment started with eucalyptus leaves to be collected and from a riparian tree next to an uncontaminated site in the Koto River. And then those eucalyptus leaves were deployed in that site for 4 weeks to be colonized by microbes. And this is a process called conditioning. After 4 weeks, conditioned leaves, most abundant shredding invertebrates, mayflies, and river water were collected from that undisturbed site in the Koto River. They have been transferred to transported to the lab. Experiments were carried out in 200 milliliter glass vials and involved exposing the leaf litter to each of the 4 pharmaceutically active compounds separately at 3 concentrations. 1 times ERC, and that's the environmentally recorded concentration. So for each pharmaceutical, we have identified the environmentally recorded concentration, and then we expose those conditioned leaves to that concentration, and then to double that concentration, that's the 2 times ERC, and triple that concentration, so 3 times the ERC. With and without shredding mayflies, so we had 2 groups. Each group, one has mayflies or shredder in them, the other group without the shredders, and incubated in an environmental chamber for 4 weeks. Treatments, including control, were set up in 24 replicates. Each group had 12 replicates for each treatment. So each treatment had 12 replicates. That's 12 with shredder and 12 without shredder. I have a note here that in pharmaceutically active compounds, concentrations in wastewater treatment plant discharge tend to be flow dependent, and prolonged exposure at the reported level was not realistic for real world conditions. Our treatment was designed to mimic a natural event where wastewater treatment plant discharges a single pulse of pharmaceuticals, for example, as a consequence of rainfall event. Consequently, we did not measure nor seek to maintain any pharmaceutical concentration throughout the duration of the experiment. During incubation time, the fluorescence intensity for 3 extracellular enzymes, beta-glucosidase, leucine AP, and phosphatase obtained using fluoroscan. Mass loss for each leaf obtained by measuring the decomposition rate constant, K, according to the equation you see in front of you. For the enzymes, the extracellular enzymes, the activity was measured using the equation that you see also at the bottom there for the enzyme activity. This is a conceptual design of the experiment. And this is the setup of the experiment. And here, as you see, for each pharmaceutical, we have shredder absent, shredder present. So that's 2 groups. And then for each group, we have 3 different concentrations. That's 1 times and 2 times and 3 times the environmentally recorded concentrations. We have the controls as well. And all times 12, that's 12 replicates for each treatment. And now we are got to the results. And as you see here, so A is for amoxicillin, B is caffeine, C is paracetamol, and D for sertraline. So with no shredder, all 4 pharmaceutically active compounds reduce the leaf litter decomposition rates, particularly at high concentrations, as you see on the y-axis. At high concentrations, that's very clear. So the no shredder case is represented by the black triangles, and that's the mean and standard errors for the no shredders. And with shredder are the hollow circles and standard errors. So those, if you look at the no shredder, so those black triangles, and as you see, the effect appear clearly at high concentrations. Sometimes at 2 times the concentration or 3 times the concentration. With shredder presence, leaf decomposition rates were lower for all pharmaceutical treatments except caffeine. And that's B, the figure B, if you look at it. Except caffeine, you see that at 3 times the concentration, caffeine had significantly increased the decomposition rates. And that was opposite to all the others. If we look at the enzymes, the extracellular enzymes activity, we can see that the extracellular enzymes beta-glucosidase and leucine AP showed reduced activity in the presence of all 4 pharmaceuticals at high concentrations. I'm not going to go through all of them, so I just picked the amoxicillin one, and that can be more clear here on the amoxicillin. So you see at high concentrations for the top ones, the top one is beta-glucosidase and the middle one is leucine AP. And for them, at 2 times and 3 times the concentrations, their activity has been reduced. This didn't happen to the last one, the last triangle, which is the phosphatase. In fact, at higher concentration, phosphatase increased. So we come to the conclusion of this chapter. Leaf litter decomposition rates are affected by the 4 pharmaceutically active compounds, especially at high concentrations. In the presence of a shredding invertebrate, leaf breakdown rates reduced when exposed to amoxicillin, paracetamol, and sertraline, but oppositely increased when exposed to caffeine. And now, from there, we had to think about the real life. In reality, these pharmaceuticals, they don't go into the streams one by one, but they go there into mixtures. And that's why we had to go to the next step and study them in mixtures. And this is what we did in chapter 5. which is titled The Effects of Multiple Pharmaceutically Active Compounds on Leaf Litter Decomposition Rates in Streams. This chapter focused on answering the question, what are the effects of pharmaceutically active compounds, stressors in combination on leaf litter decomposition rates? So when we have them together. The aim of this study was to understand what type of effect on leaf litter decomposition rates and extracellular enzymes each mixture will exhibit, because we have different scenarios of mixtures here. And it's not one mixture. We prepared or designed a different scenarios of mixtures using the same for pharmaceuticals we used in chapter 4. So we wanted to understand what type of effect do they have on leaf litter decomposition rates. Is it going to be an additive effect, which means the combined effect of multiple stressors is equal to the sum of the effects of single stressors? Or is it going to be a synergistic effect, and that's the combined effect of multiple stressors is more than the sum of their single effects? Or this might be just an antagonistic effect, where the combined effect of multiple stressors is less than the sum of the effects of each stressor alone. These types of effects, actually, I remember Ben Kiefer gave a very good talk about them a couple of months ago, I guess. And this is what we really wanted to know in this chapter. So the method I used was conditioned leaves, invertebrate shredders, stream water were collected as the same way in chapter 4. So I'm not going to go through the details of that again. 4 scenarios of mixtures have been designed: paracetamol and sertraline. So we mixed paracetamol and sertraline at all 3 different concentrations. The same way, the concentrations are the same concentrations we reported in the previous chapter. Caffeine and paracetamol, that was another mixture. Paracetamol and amoxicillin, another mixture. And then we mixed all 4 pharmaceuticals, amoxicillin, caffeine, paracetamol, and sertraline in one mixture at all 3 different concentrations. So as I said, the pharmaceutically active compounds mixtures were prepared in 3 concentrations: 1 times the ERC, environmentally recorded concentration, 2 times the ERC, and 3 times the ERC. The experiment was carried out in 200 milliglass vials and involved exposing the leaves to each of the pharmaceutically active compounds mixtures with and without invertebrate shredder, but this time for 42 days. 12 replicates for each treatment. Mass loss was calculated using the same equation we used in chapter 4, and the same thing for the extracellular enzymes. We used the same equation. Of course, we are not going to invent a new equation for each chapter. And if we look at the results here, with no shredder, most pharmaceutically active compounds mixtures had no significant effect on the leaf litter decomposition rates, except for paracetamol amoxicillin mixture, which reduced slightly the leaf litter decomposition rate at high concentration, as you see there in the bottom. In the case of with shredder, again, most mixtures had no significant effects on leaf litter decomposition rates, except the caffeine paracetamol mixture, which increased the leaf litter decomposition rates at high concentrations. None of the 4 mixtures, if we look at the extracellular enzymes here, none of them had significant effects on beta-glucosidase, leucine AAP, or phosphatase, suggesting that the paracetamol amoxicillin mixture and caffeine paracetamol mixtures may affect enzymes other than the ones included in this study. Here, this is an illustration of the differences in effects on LLDRs between single pharmaceutically active compound and various mixtures of the same pharmaceutically active compounds. At 3 concentrations, 1 times, 2 times, 3 times, as you see on the left-hand side. Pharmaceutically active compounds mixtures exhibited fewer effects on leaf litter decomposition rates compared to each pharmaceutically active compound alone. The differences were determined based on the calculated decomposition rates and then represented in terms of colors. Green and red colors indicate increase and decrease in the decomposition rate. So green is an increase in the decomposition rate, and red color is the decrease in the decomposition rate. Light colors represent slight strength of the effect. Dark colors represent a strong effect. Boxes with the dash sign represent no effect, and at low concentrations, that one times, that's the environmentally recorded concentration, no significant differences have been observed between the single and mixture effects of the pharmaceutically active compounds, but at higher concentrations, 2 times and 3 times, the effects of all pharmaceutically active compounds mixtures has been weakened and antagonized. Antagonism, I think, as you see, is dominant, because and just to illustrate that more and explain it a bit more, I just picked the caffeine case here. As you see, if we look at the caffeine here, you will see that at 3 times the concentration, caffeine highly significantly increased the leaf litter decomposition rate. That's when caffeine was alone. But when caffeine was in a mixture, although it has increased the leaf litter decomposition rate, yet the increase was less than the one when caffeine present alone. So, and you can see that from the difference in the colors. So the darker green color shows a higher or more significant increase in the decomposition rate. And that means caffeine has been weakened here compared, or the effect of caffeine has been weakened in mixture compared to its effect alone. So we get here to the conclusion of chapter 5. Most of the combinations had non-significant effects on leaf litter decomposition rates or extracellular enzymes. A comparison between the results in chapter 4 with those in chapter 5 indicated that in most cases, the effects of pharmaceutically active compounds mixtures were less than those of the single pharmaceutical. This suggests that antagonistic interactions were dominant and there was no evidence for additive or synergistic effects. Relief? Yes, of course, big relief. After I saw the single effect of each pharmaceutical in chapter 4, I thought that having more pharmaceutically active compounds together or mixed together may increase the effect and cause more problems. But unexpectedly, the mix of the pharmaceuticals tend in most cases to cancel each other effects. And that, of course, is a good reason to be relieved, because those chemicals in reality enter freshwater mixtures, not one by one, but they enter in mixtures. They get in together. In 2018, Aaron Richman and Emma Marshall reported the detection of about 69 pharmaceutically active compounds in streams near Melbourne. But I hope, after this finding, actually, I hope they may cancel each other effects, just likewise the findings here. 2004, I was relieved as finally my father was able to make it to Australia. A huge stress fell off my shoulders, but that didn't go well for long. A few years later, my father had a stroke, and I went again under stress, thinking of his new medical condition. So one stressor go, another stressor come. This might be the case when we look at pharmaceuticals' impact on freshwaters. Maybe multiple pharmaceutically active compounds cancel each other effect, are accompanied by different stressor. For example, an environmental stressor. In the time of climate change, global warming was the environment stressor to think about. And that takes us to chapter 6. Effects of multiple pharmaceutically active compounds and temperature on leaf litter decomposition rates. This chapter focused on answering the question, what are the effects of pharmaceutically active compound stressors in combination with a temperature stressor on leaf litter decomposition rates? And the method was conditioned leaves, invertebrate, stream water were collected the same way as previous two experiments from Cotter River. Experiments were carried out in 200 milligram glass vials, exposing the leaf litter to amoxicillin alone and to a mixture of the 4 pharmaceutically active compounds, with and without the mayfly shredder. So leaf litter decomposition rates were exposed to amoxicillin alone to test the method validity. And we found the method to be reproducible. So I'm not gonna talk about the results came out of the exposure to amoxicillin because it is quite similar to the results we had in chapter 4. And I will just focus on the results from the mixture of the pharmaceutically active compounds. Continue with the method. The pharmaceutically active compounds mixture was prepared in 3 concentrations, 1 times the ERC, 2 times the ERC, and 3 times the ERC. Experiments were carried out, and this is the new entity now we have in this experiment, carried out at 3 temperatures, 20, 22.5, and 25 degrees Celsius. Temperature treatments were based on the NARCLIME Australia climate change projection for the ACT region, 12 replicates for each treatment. And here is the design that you see for this experiment. It's quite similar experiment setup as in chapter 4 and 5, but this time I used only 1 mixture scenario, mixing all 4 pharmaceutically active compounds and the exposure to pharmaceuticals run under various temperatures. You see the temperatures there on the top, and we used, as I said, the NARCLIME Australia climate change projection for the ACT. So if we go to the results, as we see here, in the case of no shredder, the no shredder actually is the first one, A, and the with shredder is B. So in the case of no shredder at ambient temperature, the blue bars, those are the ambient. When I say ambient, that's the 20, which is the lowest temperature, which is the current ambient temperature in the projection. At that temperature, no significant effects has been observed on the decomposition rates. But as the temperature and concentrations increased, we started to see significant reduction in the leaf litter decomposition rates. And you can see that at 3 times the concentration and with the green and yellow bars. Here, there is an increase in temperature and increase in concentration as well. So when they are combined together, there is an effect. So remember that this is the case of all 4 pharmaceutically active compounds together, mixed together. And when we test them at ambient temperature, they had no significant effect at all. But here, the increase in temperature, along with the increase in the concentrations, showed us a significant effect on the decomposition rates. If we look at, in the case of with shredder, we see the effect only at the highest temperature and highest concentration, which is 3 times the concentration. And let's have a look at the extracellular enzymes here. Yep. So for the extracellular enzymes, I didn't bring all of them just in the sake of save time. I just brought the phosphatase, And as you see, with or without shredder, no significant effect has been observed on phosphatase activity at low or medium concentrations or temperatures. But in the case of with shredder, which is F, we notice a significant reduction in the activity at 3 times the concentration and the highest temperature of 25 degrees Celsius. And that takes us now to the conclusion of this chapter. The study shows complex interactions between pharmaceutically active compounds and temperature, suggesting the potential for significant changes in freshwater ecosystems affected by these stresses in combination. Reductions in decomposition rates may reduce energy flow to higher trophic levels, with consequences for fisheries, productivity, and rates of nutrients turnover. The study highlights the need for more detailed studies of pharmacokinetics and the range of temperatures and in the presence of other chemical stresses. In an applied sense, it shows the urgent need to incorporate climate change as a factor in determining risks to the freshwater ecosystems processes of discharges of a range of chemicals, including pharmaceutically active compounds. Now, an overall research conclusion. This research contributes to the effort to understand the likely ecological effects of multiple pharmaceutical stressors on freshwater ecosystems. It is evident from this study that pharmaceuticals and personal care products can have biological impacts on freshwater ecosystems. And this is in response to CTAP question. With increasing evidence of pharmaceutically active compounds, residues in rivers and streams, this research suggests that there is basis for substantial concern about the biological impacts of pharmaceutically active compounds in the environment. The research provides critical foundational knowledge for future investigations of pharmaceutically active compounds, contaminants in fresh waters. The findings of this thesis will be of considerable benefit to freshwater ecosystem and resource managers, policymakers, and regulators. The information provided will enable them to form the most effective policies and devise ways to enact stricter water quality guidelines. How? Based on the findings, water resources managers and policymakers can develop more efficient wastewater treatment methods to remove pharmaceutically active compounds residues before discharged into fresh waters. Encourage, support, and fund researches that study pharmaceutically active compounds' effects on freshwater ecosystems. Shape and enact policies that encourage pharmaceutical manufacturers to develop methods that produce more environmentally friendly medicines. The findings of this research actually can be used to create models to estimate the size of change in vital freshwater ecosystems processes when exposed to pharmaceutically active compounds. This can be achieved by applying long-term monitoring strategies accompanied by ecological theories and information on ecosystem resilience to change by pharmaceuticals pollution. Future research opportunities. Based on the complex interactions between different pharmaceutically active compounds, research is needed on broader range of concentrations and wider range of pharmaceutically active compounds, studies which maintains constant concentrations of pharmaceutically active compounds across the duration of the whole experiment. Study the possible effects of chiral pharmaceutically active compounds on freshwater ecosystems functions, both individually and in conjunction with other achiral and chiral compounds And a potential question for that is, how do their effects differ from those of racemic pharmaceutically active compounds? So I am actually from chemistry. I have a chemistry background, and I believe that some of you maybe don't understand some of these terminologies like chiral and racemic. So what is a chiral compound? They are molecules that have the same chemical elements and in the same empirical formula, but they are non-superimposable mirror images of each other with different orientation are said to be chiral. One of the most famous examples of chiral object is the hand, the human hands. So your hands, they are mirror image to each other, but they are non-superimposable. So they are chiral. They are known as chiral. So what we have actually when pharmaceuticals, in chemistry, when a molecule comes pure with one, the compound comes pure with one molecule only, that molecule called enantiomer. But in reality, when pharmaceuticals are manufactured, most of them come out with both enantiomers. The both mirror images. So they have 50% of this compound or molecule and 50% of the other molecule all together in one tablet. So that tablet you take, whether it's Panadol or whatever, it has both enantiomers in it, the two mirror images. And these two, in this case, when the pharmaceutical has both enantiomers, it is called racemic. So it is not a pure compound. It's called racemic. So the pure compound come mostly with one enantiomer. I can't say 100% one enantiomer, but in medications like Nexium, for example, up to 90% pure enantiomer. So future researchers need to look at that and study all mixtures cases, including racemic, that comes with the two enantiomers, and pure molecular compounds that come with only one enantiomer, and put them in the right design to do that. Also, for future research, the inclusion of pharmaceutically active compounds metabolites in the mixture scenarios. That's very important, because most of the parent compounds dissolve or break down to their metabolites, or maybe even their metabolites are what we find actually in the streams when we do these sort of research to detect pharmaceuticals in streams. Also study the effect of pharmaceutically active compounds mixtures in the presence of other natural and anthropogenic stressors, for example, storms, drought, excessive nutrient levels, pesticide pollution, and land use, in case studies that mimic real situations. Repeat the studies in this research under more field-based conditions. And finally, study other responses to the 4 common pharmaceutically active compounds that have not been addressed in this research. For example, studying the activities of other extracellular enzymes, because we studied only 3 enzymes, beta-glucosidase, leucine AP, and phosphatase, but there are many other enzymes released and excreted during the decomposition process. So they can study the effect on the activities of those other enzymes. Also the shredder behavior and ergosterol as an indicator of fungi mass, because those microbes that colonize and condition leaves mainly containing fungi, bacteria, and maybe other type of aquatic microbes. So that will be good to do all these sort of studies in the future, and will enrich, of course, the knowledge and library that trying to be filled with all the required information. So the right policies and the best management of our freshwater ecosystems can be achieved. At the end of this, I have 3 messages to take home. The first message is whenever you take a medicine, think about its fate, where it goes, how it degrades, and how it affects the environment, mainly the freshwater ecosystems. Think about it. Don't just go and take that. I felt a headache, I had a headache, just take the Panadol straight away without think about these things. My second message is to the PhD students. You may notice that I was telling some important events in my life while telling the project story. I wanted to tell you that we all come under various stresses, but that doesn't mean we give up and quit the research we are doing. If any of you think about something like that, just remember the guy who had many stresses and obstacles, but at the end managed to make it at least to the final seminar talk. My last message goes to the supervisors. A brief email support from you would greatly encourage and strengthen your students. Please don't ever underestimate your role in changing and restructuring the life of your students. And from here, I may take the opportunity to acknowledge my most supportive figures during my PhD journey, my supervisors, Fiona Dyer and Ross Thompson. Thank you so much for your support, uphold, guidance, and really useful advice. Thank you so much for your patience. You've been so patient with me and showed a high level of understanding to my circumstances. Whatever I say or do, I won't be able to pay you back. I look forward to work with you in the future, whether I'm here in Canberra, Melbourne, or maybe on Mars. And don't worry, I'll find a way to contact you from Mars. Ben Kiefert, thank you for your beautiful advices and for allowing me to use your environmental incubators. Ashraf Ghanem was the first person to introduce me to the fantastic community of the University of Canberra. Thank you so much, Ashraf, for that. John Bray and Alex Henderson, unfortunately, I don't have a photo of it. I do thank them for their help and support in the field. I also acknowledge Anthony Davidson for his assistance in the data analysis and related statistics. Thank you a lot, Anthony, and thanks for teaching me how to use R, the R program. Thanks a lot for that. Simon Foster, Janine, Barbara, Lara, all of you, thank you so much. In fact, I have a long list of names to thank that may take another 40 minutes. But because of the time limit, I will say my acknowledgement and appreciation to the IAE, COWS, and the University of Canberra members, students, and staff, as deep as the ocean and as wide as the sky. Thank you all.
Thank you, Haysem, and I think you will all agree that that's a wonderful story, both of research and of life. Haysem's going to join you now live to ask you some of his questions. We've recorded this in the depth of night while Haysem's new baby sleeps, but I will stop the playback right now, and you will now have a real live Haysem to talk to ask your questions either through chat or through the microphone. So I'll stop the recording and pass over to Haysem live.
Hi, I'm with you guys, yep.
Okay, so we're back live. So we are running a little bit late on time, but we have time for 2 or 3 questions. So can we have any questions for Haysem? So question in the chat, Haysem, is caffeine a pharmaceutical, and to what type of drug classification does it belong?
All right, yeah. Thanks for that question. Actually, I've been asked that question in many of the conferences I attended before. Yes, caffeine it is a pharmaceutical, and it belongs to the stimulant therapeutic class. In fact, lots of people think caffeine is only in the beverages we drink, hot or cold, and that is correct, but it is also a pharmaceutical, and it is found in many brands. anodine, which is given for headache and migraine, Beckmans for influenza, Lamicin, Panadol. Panadol has actually 500 paracetamol milligrams, but also it has, or in its active ingredients, about 65 milligrams of caffeine. Solpadeine, Sodafine, lots of brands, actually, I can count them. There are lots of brands that contain caffeine in them as an active ingredient. So, by the way, caffeine and cocaine are cousins. They both stimulants, but one is illegal, that's cocaine, and the other is still legal until now. So I have no idea what's going to happen after this talk. So many of you, I wish you enjoy your coffee because nobody knows what's going to happen tomorrow.
I think if you banned caffeine during lockdown, Haysem, there would be an uprising. I don't think that's likely to happen anytime too soon.
I love coffee, actually, and I don't want to do that. Yes.
I have a question for Ben, from Ben Cafford.
All right, yeah.
Thank you, Haysem. Great talk, and thank you. I've got a couple of questions. I wasn't quite sure why you concluded that the combined effects was antagonistic, particularly when you have like caffeine increasing the breakdown and then other things decreasing it. Well, it's possible, I'm not saying it's not antagonist, but it's possible you add those two together and that gets no effect. But also, even if that doesn't happen, if there's no effect of the multiple compounds, then I'm not quite sure it falls into the categories of antagonism and synergistic or additive. It's just that it's having no effect. So, and the other thing I wanted to ask, if there's time, is just with the temperature ones. That was with constant temperature. The temperature was always at 20 or 22.5 or 25, or was it fluctuating with the daily cycle? And how do you think this would happen in the real world, where you do have daily cycles which could very well be going between 20 and 25 within a day? So bring in a change, what you think might happen there.
Yeah. All right. Yeah. Thank you, Ben, for your question. Why I did conclude that antagonism, or most of the cases were antagonistic, the effects were antagonistic. The issue is, I didn't say all of them, I said most. So antagonism, if we look at most of these mixtures, and if we can bring that, Ross, the comparison between chapter 3 and 4, that's slide 31.
No, we can't do that. We're not.
All right. Okay. So if we look at most of the mixtures, they showed no effect. And that's antagonism for each single one of them, whether it's increasing the effect or decreasing the effect. When there is no effect, that means the effect has been reduced. So that's why I considered that as an antagonism. Of course, in the case of caffeine, paracetamol, we know that paracetamol reduce reduce the decomposition rates, while caffeine alone increase it. When we mix them, we saw an increase. That means caffeine effect was dominant. So, but still that increase was less than when caffeine was alone, or present alone. So that's an antagonism in the caffeine effect. For the paracetamol, by the way, it is more like a reverse effect. So that's completely, different effect from antagonism or synergistic or additive. That's the opposite way. So because we saw an increase now when paracetamol was in this mixture. So yeah, more details will be in the chapter. If that chapter got the chance to be published, yeah, you will see that in the paper with more details on it. If I go to your other question about the temperature, yes, I actually, it was constant. So 20 for the whole duration of the experiment for one, 22.5 for the other, 25 degrees for the third one. And that was constant. We didn't do the fluctuation. And guess what? We intend this research actually to be benefited from worldwide. So there are countries where these sort of temperatures are constant. Constant, I mean, most of the day, not all the day, but most of the day, or maybe day and night. Some areas in Asia and Africa, trust me, they get that 25 for the whole day and night, and even worse than that. So yeah, so also I put in the limitations that in the future research, maybe more variables will be looked at. So that fluctuation you're talking about regarding the temperature can be looked at in future research.
So we will go to one last question from Madeline in just a second. I noticed that Alika has put in the chat the return med site. So one of the things that's arisen out of concern over pharmaceuticals and the environment has been a program funded by the Commonwealth government to encourage people to return medications for reuse and recycling, as opposed to emptying them down the toilet or down the drain, which is what people have done in the past, and that can cause really large peaks in pharmaceuticals. So thanks, Alika, for that. That's one of the really positive things that's arisen in the last year or so, really, from knowledge about these processes. Okay, one last question, and from Madeline.
Right, hello.
Hi, Madeline.
Hello. I wanted to ask, so with these shredders that are like helping decompose the leaf material, is it possible that over a long time of being exposed to these pharmaceutically active compounds, that they would build up a tolerance, and would that affect like the data that you're looking at?
Yes, good question. Yes, that's possible. That's possible, and this is one of the things that we did not actually looked at in our research, but I encourage that to be happening in the future researches. Actually, I put it in the limitations, and I put it also in the future research for future researchers to look at it at that by exposing those invertebrates for longer time to the pharmaceuticals we studied. So yeah, that's something to be looked at if they may build some sort of tolerance. So it is a concerning thing, yeah. And we know even not only the shredders, even the microbes. The microbes, we know the bacteria resistance, that we have. It's something, it's a big concern actually at the moment, where many bacteria has been able to tolerate antimicrobial drugs or agents or compounds. And so it's something that we definitely going to look at in the future.
Okay, well, we are at the end of our time, and we are very glad that there wasn't the sound of a crying 2-week-old in the background for Haysem now, so that's a triumph in its own right. Well done, Haysem, for all you've achieved. Congratulations. Please join with me in congratulating Haysem on this achievement, and we'll look forward to hallway conversations as follow-ups when we can all be back in the same hallways again. So well done, Haysem.
Thank you so much. Thank you all. Yeah. Thanks a lot.
I can hear that. There and right. Catch up with you for the next IAE seminar. See you, folks.
Thank you. All right. Thanks, Ross. Thank you, Fiona. Yep. Thank you all.